Great, thank you so much for having me. So neuroimaging in long COVID, what we're looking for is cognitive dysfunction. A lot of patients have brain fog, cognitive dysfunction, and how do we explain that using neuroimaging? So previous studies have shown that reduced perfusion in the frontal, parietal, and temporal lobes may be involved. Other fMRI studies have demonstrated altered functional connections in the brain. And then the cognitive dysfunction we're talking about typically involves episodic memory deficits, as well as impaired attention, working memory, processing speed, and verbal fluency. So my hypothesis is that on structural MRI of the brain, we will demonstrate alterations in gray matter and cerebral blood flow in regions aligning with patients' symptoms. That's my goal. All right, so our methods, I've been enrolling patients in our study since 2022, and we're still enrolling. We have 32 individuals with new cognitive dysfunction lasting more than 3 months after COVID, and then 22 individuals who fully recovered from COVID. The two groups are matched for age, sex, race, and ethnicity, and we do have exclusion criteria. So our sequences that we're going to talk about here are T1 structural, FLAIR looking at hyperintensities, arterial spin labeling, which is the perfusion scan, and then we used a couple of post-processing techniques to get the quantitative data, and our statistical analysis was adjusted for multiple comparisons. So our study groups, the long COVID versus controls, across age, sex, race, time since COVID, and vascular risk factors, there were no significant differences. And in terms of symptoms, a requirement of the study was to have a cognitive dysfunction, a cognitive symptom. So all participants had a cognitive symptom. And then also there was a high prevalence of autonomic symptoms, fatigability, post-exertional malaise, dysregulated heart rate, as well as mental health, anxiety, depression, apathy, difficulty sleeping symptoms. So first looking at cortical gray matter volume, this is the actual volume of the brain, which decreases as we age naturally and decreases at an accelerated rate in certain conditions like MS or Alzheimer's. So we're looking for this in long COVID. We can see here above the top dotted red line that there are several regions where individuals with long COVID demonstrate decreased cortical gray matter volume. So I'll go through those. So one of the most interesting findings, the superior frontal gyrus, which supports speech initiation, impulse control, behavioral flexibility, and executive function. I've had a lot of patients tell me they can no longer multitask. Broca's area is essential for phonological syntax and semantic language processing, so language meaning. The isthmus links the parahippocampus and the posterior cingulate, which is a very key memory circuit. And we have a lot of patients forget full conversations, forget how they would return to their job in nursing, say, and forget their protocols. And then also that sensory emotional integration. And then the supramarginal gyrus, which has shown up again and again in a lot of our sequence, supports phonological retrieval, so that word finding difficulty, and reading within the posterior language network. And I've had a lot of patients with difficulty reading a book and not be able to read for school, for work, for pleasure is something that they really love doing. So many of the affected regions affect language, memory, and sensory systems. So next, I was very interested in looking in the autonomic phenotype, which was a large portion of this cohort, and there were several regions that were affected. So again, the isthmus came up, and the prefrontal cortex, and both of these structures are part of the limbic system, which is responsible for emotional regulation. And there were also, and here was the difference is these were higher volumes, which is very interesting. So those with the autonomic phenotype had possibly more activation of these circuits. And then the orbitofrontal cortex regulates autonomic responses such as heart rate and blood pressure. And that was also increased in patients with autonomic dysfunction. So next, I honed in on the neuropsychological testing, so the cognitive testing, and there were many regions that associated with reduced cortical gray matter volume. So the precentral gyrus contributes again to the verbal learning, speech production, particularly motor speech, and processing. So additionally, the lingual regions convert verbal information, so this is between like the occipital lobe where you have vision, and the parietal lobe convert verbal information into visual representations that support learning and memory. And then the superior frontal gyrus supports strategic verbal memory processing. And all of these regions were reduced in volume. All right, so next I'll go into cerebral blood flow. I was particularly interested in this given the previous studies, seeing if we could replicate this in our cohort. And overall, this is adjusted as we've added more participants at this time. There is no difference in perfusion between the groups when adjusting for multiple comparisons, but we're still enrolling. But I still wanted to dive deeper to see if this related to any of our patients' symptoms. So the autonomic phenotype had many associated regions with hyper and hypoperfusion. So again, the superior frontal gyrus and the precentral gyrus. Superior frontal gyrus is key for sympathetic and emotional regulation, so your reaction to stress. And then the precentral gyrus is involved in motor-driven autonomic modulation, and these demonstrated hypoperfusion, and that's basically your reaction to exercise when you start exercising. Specifically, dysregulated sweating was associated with hyperperfusion in the parietal operculum, which mediates temperature, sensation, and discrimination. And then fatigue was associated with globus pallidus hyperperfusion, which is a key area for reward and motivation. And this is consistent with basal ganglia alterations seen in chronic fatigue. So underscoring the overlap between these conditions. All right, so in terms of neuropsychological testing in cerebral blood flow and perfusion, there were several regions that were affected, but the most interesting was the cognitive failures questionnaire. So it's a questionnaire that asks patients about their daily life and the lapses that they're experiencing in perception, memory, motor, their daily life, and that was actually the strongest finding and linked with several regions. In the brain, so the patient's perception of their dysfunction is correlating with decreased perfusion in key areas of the brain. Those who tested poor on verbal memory demonstrated cerebellar hypoperfusion, and those who tested poor on verbal learning had hypoperfusion in the temporal fusiform gyrus. So the cerebellum and the temporal lobes key in these circuits. And so in summary, the cortical gray matter, we saw reduced volumes in regions critical for language, memory, and sensory processing. Increased cortical gray matter volumes appeared in areas linked to limbic function and autonomic systems, which we know may be working on overdrive. Cerebral blood flow, there were no overall group level perfusion differences, but the autonomic phenotype showed hyperperfusion in areas related to the central autonomic network and those supporting sympathetic, emotional, and activity-related regulation. And then cognitive symptoms correlated with hypoperfusion in the frontal, temporal, and parietal lobes. All right, I did it in time. Huge thank you to my patients and our dedicated participants who we've learned so much from, and a big thank you to the COVID Mind study at Yale and our collaborators. All right.